Hello, everybody, and thank you so much for tuning in. My name is Esme, and I'm a producer for the How To Academy. And joining us this evening is Professor Beth Shapiro. Beth is Professor of Ecology and Evolutionary Biology at UC Santa Cruz. She was also awarded the MacArthur Genius Grant, and she's a National Geographic Explorer. Tonight, she joins us to mark the release of her new book, Life as We Made It. From the first dog to designer pigs, Beth will be exploring human intervention in the natural world and showing us that it isn't as recent a phenomenon as we might think. Beth's going to speak for around 50 minutes, and then it will be over to you to ask questions. So please send any thoughts, queries, anything at all into the chat, into the Q&A, sorry, not the chat, and we'll try and get through as many as we can. So without further ado, please join me in giving a very warm welcome from all the way across the pond to Beth Shapiro.
Thank you so much. Here, I'm going to start sharing my screen to try to make this work here. And let's go. All right, how's that look?
Perfect.
Good? All right, so thanks so much for joining me today. It's 10 o'clock in the morning where I am, so that's kind of easy and straightforward. I know it's later in the day for you, but I've had a couple of cups of coffee, so hopefully I am ready to go. Today, I'm going to talk to you about how to supercharge conservation. So as Mika said in my introduction, I'm actually a scientist in my day job anyway. I work in a field called ancient DNA. This is basically an area of research where people like me develop methods to try to recover and analyze DNA that's preserved in the remains of plants and animals that used to be alive. It's not a very old field as far as scientific fields go. The first successful recovery of ancient DNA was in 1984, when scientists working in Alan Wilson's lab at UC Berkeley managed to recover short fragments of DNA from the preserved skin of this guy, a quagga. The results were not all that remarkable. It turns out that a quagga is, as looking at this picture might lead you to believe, a type of zebra. But the idea that DNA stuck around and didn't degrade right away after death was remarkable. And in the nearly 30 years since that time, we've learned that DNA is actually preserved in lots of things, like bone and teeth and hair, and even mummified bodies, like this little very old, about 35,000-year-old extinct cave lion, and these birds down here. Also poop and shells and plant material and things like these moccasins, which are from Promontory Cave in Utah, where we can get DNA both from the leather here to figure out what sorts of animals people were using to make the moccasins that they were wearing, and also from the inside, where we can learn about the people who were wearing these moccasins. To date, the oldest bit of DNA that's ever been recovered is from this mammoth tooth on the lower left-hand corner of the slide. I don't know if you can see my marker here, but this is from a tooth of a mammoth that's known as the Krestovka mammoth. This tooth is around 1.2 million years old, and earlier this year we published a genome from this mammoth that we extracted from this tooth. This is really old, and DNA of this age is super rare. For the most part, DNA tends to degrade into pieces that are too small for us to use after about 50 or 60,000 years, and only in the very best environments. So over time, we've learned lots of cool things. The very first bit of ancient DNA research that I was involved with was with the dodo. I was a PhD student at Oxford, lived in the UK for almost 10 years, and not all of them during my PhD, I wasn't that slow. But one of my first ancient DNA studies was to get a little chunk of bone from the leg of the famous Oxford dodo and show that it is in fact a type of pigeon, which was surprising and a little bit disappointing to bunches of us, I guess. We thought it was going to be something way more spectacular than a pigeon, but it's still pretty spectacular. We've also learned since then, studying bones from the North American Arctic and Siberian Arctic, that there were at least 2 different species of horse-like animals that were really undescribed from a paleontological perspective. And what's probably the most famous ancient DNA study showed that this little pinky bone, if you see this little bone right here, was used by scientists at the Max Planck Institute in Leipzig to characterize a new species of human that people didn't know existed, the Denisovans, which were closely related to Neanderthals. And the genomic data from Denisovans and Neanderthals was used to show that our ancestors, when they moved out of Africa and started spreading across the world, actually interbred with both Neanderthals and Denisovans, and that all of us are descendants from these hybrid Neanderthals and anatomically modern humans. But getting back to this the very beginning of the field of ancient DNA, we also see that whenever we publish anything in ancient DNA, there is one question that all of us are asked. And Alan Wilson, here you have him, the first person to extract DNA, was also the first person to get to answer this question, that no, the possibility of actually bringing ancient species back to life is, as he says here in his quote in 1984, extremely remote. Now, nearly 30 years later, this has a name, de-extinction, and let's face it, those of you who have logged on today are here to ask me that question, aren't you? Well, fine. Here's your update, and I happen to be in a great place to update you, as I've just met last week with the founders of a new company that has the explicit goal to resurrect mammoths, and as of today, de-extinction is still not possible. We cannot bring back dinosaurs. Remember, the oldest DNA we've recovered to date is 1.2 million years old, and this is very rare, and dinosaurs have been extinct for more than 65 million years ago, unless we're talking about birds as dinosaurs. So no non-avian dinosaur DNA. Dinosaurs are not coming back to life. Very sorry. Also, we cannot still de-extinct mammoths or passenger pigeons or stellar sea cows or dodos or the Tasmanian tiger or the gastric brooding frog. But I'm just going to pause here because look at that guy. Do you see the little baby frog coming out of his mouth? This is a frog that would swallow its tadpoles and then sometime later barf up fully formed frogs. Now, if we're going to bring something recently extinct back to life, this would be my choice. I mean, how cool is that, right? And also even things like this guy. This is Lonesome George. He's a Pinta Island giant tortoise, and this picture was taken shortly before his death in 2012, by which point he had been living in a captive breeding facility for almost 40 years. He was somewhere between 80 and 100 years old when he died, and he'd spent much of his life in that facility where people had tried to breed him with closely related species, but nothing worked. All of these species are gone, and when things become extinct, they are gone forever. So why, you might ask, can we still not bring extinct species back to life? The issue is that there are technical challenges and also ecological and ethical challenges, but for now, let's stick with technical, okay? Focusing on the mammoth. We can get DNA from well-preserved mammoth bones, like this mammoth bone that my friend and colleague Love Dalen is holding here on Wrangel Island. And we can compare the genomes that we can sequence from these bones to the genomes of Asian elephants. And this is the most closely related living species of mammoths. In fact, we've done this. We know that they diverged from each other around 5 or 6 million years ago, and there are around 1.5 million differences in the DNA code between an Asian elephant and a mammoth. And we can grow Asian elephant cells in a dish in a lab, and we could consider using the tools of gene editing and genome engineering to make those 1.5 million changes, essentially transforming those Asian elephant cells growing in a dish in a lab into cells that contain a mammoth genome. Then we could find an Asian elephant surrogate maternal host and implant these cells that we had transformed into early stage embryos into these Asian elephant maternal hosts, where 2 years of pregnancy would happen and hopefully go with nothing happening, nothing going wrong. And then when that baby was born, that Asian elephant mom could teach its baby how to be a mammoth, or, well, I guess that would be difficult if the Asian elephant didn't know how to be. Actually, you know, there's so many challenges that we see here in steps 3 to 5 that if we did do this, and this is the goal of both this new company and some research teams around the world, then we would probably end up with something that was maybe just a little bit mammoth-ish. right? But, watch the space, because as I mentioned a bit ago, there's now a new company. It's called Colossal, that was formed to try to jump over these technical hurdles. It's led by tech entrepreneur Ben Lamb and George Church, a Harvard scientist who has already started editing Asian elephant cells in his lab. He's managed to make so far about 50 changes, focusing on changing genes that might help tropically adapted Asian elephants survive in the Siberian tundra. I'm sure that if I could see some of your faces, you would be shaking your heads, as I did when I initially heard of this. And I agree, this is a little bit crazy, but it is also a lot cool, because this is genuine investment in developing technology that could be really groundbreaking for conservation. As this team learn how to edit genomes from wild species, transform edited cells into living plants and animals, other species will almost certainly benefit. And while Colossal might be focusing on using the technology to bring mammoths back to life, other people can use what Colossal learned to help corals adapt to warmer oceans, make black-footed ferrets unsusceptible to plague, repopulate forests in North America with American chestnut trees, and even rid islands of invasive species like brown tree snakes that are currently eating a lot of island endemic birds. Wait, what? I'm sure you're saying. This is in the parlance of my 11-year-old who says this to me all the time. That sounds great, I'm sure some of you are thinking, but others among you are probably entirely turned off. This is crazy. Who are we to start messing with other species in this way? Shouldn't we instead back off and let nature recover? That argument posits that we have a choice between two things, doing nothing and doing all of the things. And in fact, this is the dichotomy that rules conservation today. Should we, as Ed Wilson suggests in this Pulitzer Prize-winning book, Half Earth, buy land and leave half of the earth alone? Or instead, should we get our hands increasingly dirty, as suggested by people like conservationist and environmental scientist Emma Maris? So this brings me to my two goals for today's lecture. First, to show you that we don't actually have to choose between these two things. And then, after I've done that, to show you that, in fact, we do have to choose. And right now is the time to be thinking about the choices that we're going to make in the next year and decade that are really going to have enormous consequences for conservation of biodiversity on our planet. So with that in mind, I'm going to begin by telling you a story about hippopotamuses. All right, here we go. The year was 1910. And in the United States, well, in the whole world really, but I'm going to focus for this story on the United States. There were crises. The first and most prominent crisis was a meat crisis, specifically a meat shortage, or at least super high prices for meat, which led to panic in the world around impending terribleness, even starvation. There was, in fact, conversations about eating dogs. This was blamed, at least in part, by the expansion of cereal crops across what had been the cattle ranges in the western part of the US. But in fact, the quality of the cattle ranges had deteriorated because there had been too many cattle. And by 1910, too few cattle were coming to market to satisfy demand, and people were freaking out. And that was not the only crisis of the time. In 1886, the Japanese delegation of the World's Fair gave to New Orleans, which was that year's host city, a gift, a plant that grows in the water and produces bunches of delicate, beautiful purple flowers, the water hyacinth. People loved it. They planted it in their gardens and in city parks, and then it started to grow. Patches of green, as much as doubling in size every week. By 1910, impenetrable mats of water hyacinths were choking lakes and rivers and bayous, sucking oxygen from the water and killing fish, and blocking river transportation into the Gulf of Mexico. The Department of War tried clearing it out by hand, poisoning it, even drowning it, but nothing worked. The simple but charming water hyacinth had sown an environmental and economic emergency that, as you can see here from the picture on the right, persists in many places around the world today. There was a man, however, who had a plan to solve both crises at once. Louisiana Congressman Robert Cousin Bob Broussard had crafted a resolution to be introduced to the United States House of Representatives H.R. 23261, in which he proposed to commit $50,000. That's about 7 million in today's US dollars, or 5 million pounds, to import animals into the US and stick them on lands that were not currently being used so that people could take advantage of said animals. But Broussard was not thinking about any old animal in any old place, but about importing hippos from Africa into Louisiana's bayous, where it was too wet for cattle ranching. Once there, he surmised these hippos would eat all of the water hyacinths, transforming the offending green plant into literal tons of delicious hippo meat, making everybody happy. Of course, this plan was not Broussard's originally. In fact, the idea to import African animals was originally that of Major Frederick Russell Burnham, who was a scout and adventurer, and actually the inspiration for the Boy Scouts of America. Burnham had proposed some 4 years earlier to his friends in the political elite that the US should import antelopes and giraffes and other African animals to populate the newly established land reserves in the American West. He was convinced that by bringing these magnificent and edible beasts to the United States, not only would this solve the meat crisis, but it would also foster support, in particular from sporting hunt enthusiasts, for the conservation movement. Broussard and Burnham recruited 2 others to their team, W.N. Irwin, who was actually employed by the Bureau of Plant Industry and charged with improving the country's apples, but was obsessed with solving the meat question, and Captain Fritz Black Panther Duquesne, a man of many talents and few of them legal. He was actually an accomplished scout and big game hunter. In fact, he had been employed during the Boer War to assassinate Burnham, but clearly failed since they were working together on this particular project. He was also a con artist. He'd been a German spy, a prisoner, a photographer, and a fake paralytic. Broussard actually ran into Duquesne when Duquesne was posing in a one-man show as Captain Fritz Duquesne, adept and legendary hunter of African game. Duquesne's performance, of which parts were probably true, was good enough to convince Broussard to invite him to join his panel of experts. So the 3 of them, Burnham, Irwin, and Duquesne, presented in March of 1910 their argument in favor of House Resolution 23261. The House of Congress was packed and they had lots of questions that I'm sure you have today about this, and these men tried to answer the best that they could. How might the hippos taste? Their taste might be a cross between beef and sweet pork, said our accomplished scout. Are they dangerous? WN offered that they might be dangerous if they were turned loose, but this was quickly countered by our expert con man Duquesne, who happily reported, with no supporting evidence whatsoever, that hippos were naturally tame and in fact enjoyed being led around by leash and drinking milk from a baby's bottle. It turns out people were ready for hippos in the Mississippi River. The New York Times published an editorial just after this pitch to Congress in which they declared that lake cow bacon, which I assume means hippo meat, was the next big thing and absolutely destined to solve both the meat crisis and the water hyacinth crisis at once. The stage was set for H.R. 23261 to pass and bring about a country of fuller bellies and cleaned up lakes and rivers. Now, you may have noticed that there are no hippos in Louisiana's bayous. And if you have noticed that, you might be wondering, did people come to their senses? Did they realize that hippos are dangerous and clearly no one eats them, and this is a terrible idea? This must be what happened, right? Well, if you think that, you'd be wrong. What happened was the presentation to Congress took place too late in the session to come to a vote that year. Broussard kept meaning to reintroduce the bill in later years, but never got around to it. Irwin died. Burnham was sent to Mexico to protect copper mines that were being threatened by the beginning of the Mexican Revolution. And Duquesne descended into some sort of paranoia that other people were stealing his idea to import hippos. And the bill died a silent death. Now, that's a terrible ending, you're probably thinking. A real letdown, right? So why did I tell you this story?